Hi everyone, and welcome to our webinar today. My name is Rebecca Northeast, and I'm a product manager here at Protein Tech. And so today we're going to have our webinar led to you by Dr. Lukasz Chrobok, and he's going to be talking to us about IHC and IF analysis. So briefly, I just want to start with a quick company introduction. So at Protein Tech, we have 3 core product lines through which we sell all our products. So through our Protein Tech brand, we sell our antibodies and immunoassays, and we have antibodies against 13,000 human targets, as well as over 300 ELISA kits and our IHC easy kits. Through our Humankind brand, we sell our cytokines and growth factors, and these are recombinant proteins which have been expressed in HEC293 cells. And these are excellent because this means that they have native folding and post-translational modification, meaning that they have higher activity in comparison to proteins which are expressed in, for example, bacteria. Through our Chromatech brand, we sell our nanobody-based reagents, and these are excellent for immunoprecipitation and super resolution microscopy due to the small size of the nanobody. I'd now like to introduce Dr. Lukasz Chrobok. And Lukasz is actually an old colleague and friend of mine when we both did our postdocs at the University of Manchester. Lukasz has a background in neurophysiology from the, and I'm sorry if I say this wrong, the Jagiellonian University in Krakow in Poland. He then did multiple postdocs, starting at his lab in Poland, in Krakow. Then he moved to Manchester where he met myself, followed also by a postdoc in Exeter. He now has the really prestigious fellowship, the Sir Henry Wellcome Fellowship, and he's based at the University of Bristol now. And just for such a young age and such a young scientist, he has over 16 first author papers, which is a testament to his hard work, as well as being an electrophysiology and IHC/IF expert. And so welcome, Lukasz, and I will now hand over to you.
Thank you so much, Rebecca, for a kind introduction. And yes, so to start, I have to admit my prime expertise is in electrophysiology, although throughout my research, I've been using immunostainings to answer a lot of research questions that I've encountered on my way. And I think that's a message for you as well, guys. If you are molecular biologist, electrophysiologist, or maybe you study animal behavior, sooner or later, you will all be using immunofluorescence to answer some of your questions. That's why I think this basic seminar that we host today with Protein Tech would be probably useful for you. So that's a quick agenda. I will start with a short introduction of image analysis per se. I will talk about the software that I really like to use, which is ImageJ, and its extension called FIJI. Then I'll move to basics of the theory. So I'll introduce you to types of images. I'll talk a bit about bitmaps and about binary images. Then I'll tell you a few words about image processing. So how to make your pictures look beautiful for your presentations, papers, or posters. And then the core of our seminar is to know something about measuring intensity or density of your staining in your beautiful images. All right, so the first question, an important question, is why should I even analyze my images? So, of course, most of you probably who's done immunostainings have taken these beautiful images with epifluorescent microscopes or confocal microscope, then zhuzhed it up with some software and presented them in your articles. However, I think that we should still remember that immunostainings may be a useful source of quantitative data. To have some kind of idea on the quantitative potential of your image, you need to go through a non-biased image analysis, and then on top of it, of course, perform your statistical analysis. And before you start your analysis, it is worth to mention about these 3 basic things, which can be called overall as a good scientific practice with dealing with your images, So first and the most important is, I would highly recommend you to run your experimental and control staining, and then your experimental and control imaging at the same time. I know that your pipelines are for sure standardized. You have some steps in room temperature, some steps in fridge, so 4 degrees Celsius. But still, I don't know about you, but in England, room temperature is really variable. Also, there are some other errors like pipetting errors. So it's really recommended to do your experimental and control staining at the same time. Also, it's good to blind yourselves. I know it may seem a truism, but it's really important to be non-biased during your analysis. So the easiest thing to do is to code or ask your colleague actually to code the images for you, then analyze them, not knowing what you are actually analyzing, and then again, ask your colleague to uncode it for you. And yeah, colleagues are very valuable because you can also ask them to reanalyze all of it for you again. And I know it sounds a bit laborious, but I think it's a very crucial step, because then if your analysis and their analysis is very similar, give you similar results, you can sleep well at night and know that your results are non-biased and probably true. Okay, another very important thing during your staining protocol is to include some controls. The basic controls are, for example, the usage of secondary antibody only, so you omit the primary antibody step. If you still have some signal coming from a tissue, something's clearly wrong. Also, more and more journals actually require you to do some positive and negative control. Positive control is when you stain the tissue that is well known to contain the antigen that you are interested in. And you can show that your staining positively works in this kind of tissue. Whereas the negative control would be when you stain the tissue that you are sure that the antigen of your interest is not expressed in there, is not present in there, so there is no signal. Okay, so our staining is done. Let's say our images are all blinded, coded. Then what software should we use to analyze them? My seminar today will be based on one type of software only, which is ImageJ, and its beta version, which is FIJI. So ImageJ is a free software, which I think is excellent. You can download it from the link provided here. It was developed by National Institute of Health. It's Java-based, if you feel like cracking the code. And the excellent thing about ImageJ is that it's widely used by scientific community. So if you have any problems, any questions, you can jump into some kind of forum, you can go to ResearchGate, you can probably actually find answers to your questions in the internet because probably someone else already had them. FIJI is the extension of the ImageJ. The acronym means FIJI is just ImageJ. However, it is not just ImageJ, it's much more because it contains a lot of pre-installed plugins which make this software excellent for analysis of confocal images. You can also download it for free from this link. What is important for FIJI is that if you take your pictures with most available brands, commercial brands of confocals like Leica, Nikon, or Zeiss, you don't have to export or import your images, you just open them with FIJI. So it recognizes all these formats. Moreover, it recognizes meta files. So not only your images, but also all the information surrounding your images that are provided by the file, for example, Leica file. What I mean here is FIJI will also know, for example, what is the size of your pixel, how many microns by how many microns, and all surrounding information like that. Okay, so how does FIJI look like? It's very simple, very basic, and I would love to keep this seminar today very simple and very basic. However, bear in mind, it's also a very powerful tool. So we'll do very basic things today. However, I'll try to give you some directions where you can diversify from this path, learn a lot more, and do some fancy stuff as well. So it's very simple if you want it to be basic, but also it can get very complicated if you want it to be very sophisticated. Okay, so with FIJI, you can have some, you have some tiles here. So I'll try to go through them all quickly. The first four enable you to draw region of interest. So regions at the image in the picture that you actually aim to analyze. You can draw rectangles, oval, you can draw a polygon, or if you feel very artsy, you can also do some freehand drawing. So, sorry. So basically, this looks like that. This is the, oops, so sorry. So basically, this is how the image a picture opened in FIJI look like. Here is a picture of some Dapistain nuclei, and I've just drawn some regions of interest on top of it. You can see the yellow shapes. If you want to draw more than one region of interest, you just hold Shift and draw another one. Then you can crop the picture, so you will see just the regions of interest. You can just analyze within or outside your region of interests. Okay. Oh, I'm so sorry. Something's really jumpy here. Okay, another type of region of interest selection is the tool called point measurements. So basically, point measurements are region of interest that contain only one pixel. You can draw multiple of them as well. Here I've drawn some point region of interest. You can see number 1, 2, 3, 4, 5. And then when you click Ctrl M, which is a measure option, you can see the basic measurements of these regions of interest. So you can see these number 1 and number 2 are in the object, in the nucleus. So they are brighter, they've got higher mean intensity. Number 3 is in the background, so it has a lower mean intensity. FIJI also provides you with the coordinates, so X and Y for each of these points. If you want to do some more measurements than just the mean intensity, you can of course go to Result, Set Measurements, and check whatever you like. The next tool in the FIJI panel is a line. It may be useful for some measurements. You can just draw the line and measure the distance between objects in your picture. You can also draw 2 lines and measure the angle if you need so. And the last region of interest related tool is wand. It's called a wand or magic wand. It selects the pixels of the same value. So if I click the pixel, one of these bright pixels here, it will draw around all the pixels of the same brightness and present them as a single region of interest. It's very useful for binary images that I will talk about a bit later. Also, some other basic tools. You can write some text on top of your picture. You can magnify your picture, or you can just scroll around them with this little hand. And a very, very important thing is that all of the things that I told you before, all of their region of interest, shapes and lines, we do not really draw them in the picture. We draw them on top of the picture. And this layer is called a lookup table. So this is important because whatever you do with the picture in the lookup table, you do not change any of the intrinsic parameters of the picture. That's important for your measurements. You don't want to mess with your parameters. So everything is drawn on top of the picture rather than in the picture. You can also change colors. For example, with my nuclei, they were colored in cyan, but I can change it to magenta if I want. And this will not change any of the intrinsic parameters of my picture, just because this operation will be performed in the lookup table, so on the overlay, rather than in the picture. So don't worry about changing anything about your picture if you do your operations in lookup tables. Okay, so let's move into some theory about images. So I would like to introduce you first to two types of images that you will probably work on. The first one is bitmap, which is this type that you get as a raw file, for example, from your confocal microscope. Bitmap is just a collection of pixels, and FIJI will tell you how many pixels there are in your picture. For example, here, this little cropped picture would contain 506 by 517 pixels. And as I told you before, because it is a meta file that is provided by Lukasz format, FIJI will also know and calculate it for you that this cropped image is 143.75 by whatever microns. It may be useful information, especially when you want to do some area calculations. It will give you the information about real area rather than about how many pixels are in there. That's more biologically relevant, I guess. So bitmap has a resolution. It's in 2D. It's X number of pixels by Y number of pixels. But it also has 24 bits, which is 3 channels of 8-bit. So it's a red, green, and blue channel. But most importantly, each pixel has an intensity, which is presented on the grayscale. And this intensity can vary from 0 to 255, and it is positioned on the range. So basically the pixel with the value of 0 will be super dark, pitch dark. And the pixel with the value of 255 will be just white, so the brightest possible. But you also have everything in between. Also in FIJI, you can visualize multiple color channels. That's very important for multiplex staining, which is the whole reason we do immunofluorescence staining rather than, for example, using DUB. So let's say you've got your channels stained with DAPI, then other secondary antibodies with fluorophores like Coralite 488, CY3, CY5. So you can either display all colors in one image on top of the others, or you can actually have each color as the other channel. And I will show you how it looks like. So here you can see a picture taken by me at the confocal microscope. It represents staining against GFAP, which is a marker of astrocytes. And this staining was performed using protein-tech antibodies against GFAP. And when you try to stain something that's really abundant in the cell, like GFAP, I think a nice shortcut that is also provided by protein-tech is to use primaries that are already conjugated with fluorophores, like this one. This one was anti-GFAP, conjugated with Coralite in the red channel, I guess. So an excellent protein-tech antibody, highly recommended. You can see really nice GFAP positive processes. So basically you can see astrocytes here. And then at the bottom of the picture, you can see channel bar. So if you slide the channel bar, you can see another channel, which is DAPI here. So this represents our nuclei. But with the option, when you choose image, make color, make composite, you can put all these channels on top of each other. Still, the channel bar is present here because you can still manipulate the magenta channel and the cyan channel separately. So the only thing that changed here is, again, on the lookup table, you can see them overlaid, but they're still not overlaid really. So again, you don't have to worry that when you composite them, you will lose these two channels because they're still separate. You just see them on top of each other. Okay, so with your confocal microscopy again, I'm sure you're not taking just one image, but you rather take a stack of images, which gives you this additional Z dimension, so the depth. But then before our analysis of density or intensity of our staining, we rather tend to collapse these images into the single image. So to do that, we have to use the option which is called Z project. And Z project collapses a stack of images into just one image, pixel by pixel. And it uses, it does it with few options that you can choose from. I would again recommend you to use one of these two, the average intensity or max intensity. So the average intensity projections calculates an average intensity of each pixel with the X, Y coordinates throughout the stack, so throughout the Z dimension, and then present an average intensity on your result image. The max intensity option goes through all the pixels through the Z stack in the of the same XY coordinate, chooses the most bright one, and then presents it on the result image. So coming back to our staining against GFAP and nuclei, you can see a channel bar and the Z bar. And moving Z bar left and right, we can go through the depth. However, when we do the Z project with the max intensity option, you can see that these images nicely collapsed, and you can see the nicely intense staining that is ready for your image analysis. Okay, so that was bitmap. Bitmap is good for intensity measurements because each pixel is coded on the grayscale, and it has a value from 0 to 255 and everything between in the range. The second type of picture, though, is a binary picture, binary image, which is a 1-bit bitmap. It is also collection of pixels, just like the bitmap. However, here each pixel is qualified as either an object or a background. So on the grayscale, it has a value of 0 or 255 and nothing in between. So 255 object, 0 background. It is very useful for area fraction analysis. So when you want to analyze your density of your staining, and for some fancy morphology measurements. For example, if you like to do shoal analysis, you have to binarize your images first. And of course, with both bitmaps and binary images, you can do some image processing in FIJI. It has all the options there. If you just want to make your picture beautiful, it's also a good software for that. You can change brightness, contrast, colors of each channel. You can make composite. So what I showed you before, you can transform your images to stack. You can split your stack to images. So you can have just one layer from your stack if you want to present just one layer from your stack. But again, very important thing, all of these operations are done on the lookup tables. So you change your colors, you change your brightness, you change your contrast, but this is just for your visual purposes. And if you save the picture, the image as it is, it will just be presented as an original picture. Because remember, if you do your operations on your lookup tables, you actually don't do any operations in the picture, but rather on top of the picture. So when you make your picture presentable and you actually want to save it as, for example, TIFF, you have to flatten it. And flattening, the operation called flattening, causes the lookup table to be embedded in the actual picture. So to flatten the picture, you just press the combination of Ctrl-Shift-F, or you just go to Image Flatten, and then you can save as, for example, TIFF, and that time your image will be saved as it is visible in FIJI. Okay, so back to analysis rather than making your pictures look beautiful. So I told you that with bitmaps, you can measure intensity, and with binary files, you can measure density. With bitmaps, that's easy. You just take your picture from confocal microscope, you do the Z projection, Z stack, and you image your and you measure your intensity. I'll show you how to do it in a second. But how to make your image a bitmap? So that is much more complicated. You have to go through a process called thresholding. And there are two types of thresholding. The first simple one is called global thresholding, which is just a manual selection of threshold that will qualify the intensity of our pixels as object or background. So let's say on the grayscale of 0 to 255, we decide that everything that's brighter than 150 is going to be our object, and everything darker than that is going to be our background. So just draw the threshold on the histogram of pixel intensity, and you binarize the image. So as simple as that. Actually, in FIJI, it's not a single line. It's always a mask. So it's a frame. And there are two types of masks. A red mask is a frame that you draw on the pixel intensity histogram that qualifies everything within the frame as an object. The over-under mask, on the other side, is the mask that qualifies everything inside as a background. So to visualize that, this is the red mask. You just draw them around the histogram of your pixel intensities. And okay, let's say that's an object. And with the green one, everything outside, that's the background. So this is zoom of the image of my GFAP positive processes, or just astrocytes, before binarization and after binarization. So as you can see here, I've done these point measurements. So before binarization, number 1 and number 2, the values, the mean values of pixel intensities were 122 and 7. So rather bright and rather dark. But after binarization, whatever is there in the object, it is 255. Whatever there is in the background, it is qualified as 0. So that's binary. Also remember, the color of your background or an object is the operation done on lookup table. So you can invert lookup tables, and if you just fancy, you can have your object presented as white and background as black, or just the opposite. So again, if you do the measurements, you invert your lookup tables, it will not affect the measurements. It's just for your visual purposes. You would have to flatten the image to make it forever, be there forever. Okay. The second type of thresholding, which is more complicated but unfortunately much more useful, is adaptive thresholding. So this kind of thresholding takes into account spatial variation in illumination. And why is it important? So imagine the example. For example, you are staining a brain slice that is not really homogeneous. It contains an area on the left, which is really myelinated, and area on the right, which is not really myelinated, not so. Then you perform your staining, and you can see that the myelinated area has a really high background noise, whereas the non-myelinated has a beautiful dark background, although your signal is also a bit lower. So then if you do a normal global thresholding, and you want to get rid of the background on the left side, you can also lose a lot of a signal from the right side, which is just generally darker. So adaptive thresholding is the way how to deal with this problem. Because it is a binarization which is based on context, on the local context of the neighboring pixels to each pixel if you're in your image. And it does not use the global illumination level, so the pixel intensity histogram. For these ImageJ and actually FIJI gives you some alternative algorithms that deals with the adaptive thresholding. There are 9 and that are just there in FIJI, but if you don't like them, you can of course download some more. However, I'll go through the basic ones. So again, you go to image, adjust local auto local threshold, and then this window will appear on the screen. As you can see, you can choose few things here. First, the method, which is just what algorithm you would like to try to binarize your images. First, let's do trial. So we'll see the result of all of them, and then we can pick and choose what's actually the best for our images. I know it may sound not too scientific to pick and choose, trial and error. However, with this kind of adaptive thresholding, I think this is the way to go. Some of these algorithms work perfectly on processes. Some of these algorithms work really well on round objects like nuclei, cell bodies. Some of them deal really nicely with high background intensity. And other ones are not so good with huge contrast. So when you want to try to do the adaptive thresholding at your particular staining, first, please try them all, look at them, see which one is the best, and then stick to it. Okay, then the next thing is radius. Radius is the number of pixels in the neighborhood that will be taken into account when the algorithm is making their measurements. And there are also 2 special parameters, and they depend on the algorithm used. So some of the algorithms, for example, use both of them. Some of them use none, and some of them just use parameter 1. If you want to go in depth what this parameter means for each of the algorithms, please go to the help. I will tell you about one that we're going to use, and I think it's the best. So yeah, when we try all with the radius of 15, you can see how FIJI tried to do the binarization, the adaptive thresholding. Yeah, some of them are not really good. So because these are probably not the best for processes, they would probably be better for round objects. 3 of them are quite good, but I think that Bernsen, so the first one, is actually the way to go if you want to study thin processes on the dark background. Okay, so with Bernsen adaptive thresholding algorithm, it uses radius, as all of them, and parameter 1. Parameter 1 for Bernsen is the measure of local contrast. So if your image is really nicely contrasted, you keep this parameter low. If it's not really contrasted, you're not happy with that, you've got some background, you can put this parameter high up. Again, here, trial and error is the best way to go. What I'll recommend you here is you can try to make one of your pictures, one of your images, binarized in the best possible way, set all these parameters, and then of course use the same parameters to all of your pictures that you're going to analyze, because if you change them, of course, you're going to change your results. So do a lot of trial and error, make them look beautiful, get rid of all of the background, and then stick to it. Okay, so some examples. This is Bernsen with parameters set to 1. And I just changed the radius, so the number of pixels taken into account in the algorithm calculations around each pixel. So you can see radius 5 is not the best because my asteroids are not that dense probably. So it qualified a lot of background as the actual object. Then I increased the radius and it actually looks much better. Okay, so I think I went through the basics of the theory. So I would like to sum it up before we actually do some measurements together. So there are two types of measurements that are the simplest, I guess, and they may be probably used to most of your images. So the first one is the measure of intensity. So it actually answers the question how intense your staining is, so how much antigen is there, for example, in your brain slides. But the second thing is you can also measure density. It's especially important if you measure, for example, processes, the density of your astrocytes, of your dendritic trees. But with the density measurements, we need to go through binarization, thresholding, and making our pictures binary. So measuring intensity, just bitmap, measuring density, binary images. Okay, so let's open FIJI and click it through together. All right, so yeah, this is very simple FIJI bar. I do have a set of images that I have obtained with Leica confocal microscope. So I just drag and drop, and then you can see you can choose from all the images that were in this one project. Let's use this one for our measurements. I think you will recognize this one by now. Okay, so again, this channel represents GFAP stained with the anti-GFAP correlite antibody. And the second channel represents DAPI, so it shows us our nuclei. I like to have these two channels with different colors. I code with different colors, but that's again just for visual purposes because this operation will be done on lookup table, so it will not mess with your image. Don't worry about it. So you can always change the lookup table. Let's say GFAP to magenta, and then our DAPI to cyan. Okay, so we have them both of them. Then we've got a Z bar, which just allows us to travel through the Z stack for both channels. Okay, so the first thing we want to do is we want to do Z project to analyze all the depth in one picture, in one image. So to make a Z stack, we go to image stacks, Z project. We choose the max intensity option for all of the slices, so the whole density, whole depth of our image. And here you have a beautiful image containing all these astrocytes in one picture. And still we have two channels, so DAPI and GFAP. So this is the image that is actually ready for intensity measurements. So to do our simple intensity measurements, we can just draw an oval on top of it. And then we can drag this oval across multiple ROI that we'd like to sample. I also recommend you to sample each of your bigger region of interest several times and then average the results. So to do that, we just drag it somewhere and then we press M, which is just a shortcut for measure. So you can see the mean intensity of the first measurement, second, third, fourth, and then we go to another area. First measurement, second, third, and another area, and randomly we choose the measurements. So you can see that the first three are around 90, and that's a grayscale from 0 to 255. Then another around 30, and another area is around, let's say, 10. And these results we can just put in our Excel sheet. We can do the same for all of our slices. Some of them will probably be control slices. Some of them will be experimental slices, but we do not know that at this stage because, as you remember, everything is blinded. And then, of course, we decode, unblind ourselves, and then we can put these results into further statistical analysis. But as I said, this is a very simple analysis, the analysis on the intensity. However, sometimes it is just enough. You see how simple it is to actually have some quantitative view of your slices. All right. But also, I would like to know what's the density of these astrocytes here, of these processes. So to do that, let's close these measurements. And I need to now binarize this image. So to binarize this image, I go to image, adjust, auto local threshold. I choose the Bernsen algorithm, and I know experimentally on this kind of picture that the good values for it are going to be 50 and 50, right? Now it is calculating, the algorithm is calculating its threshold in this image, which takes just a few seconds. And now it will show us the binary image. Yes. Okay, it's magenta because again, I chose it to be magenta. But if you want to make it a grayscale, you can just, yeah, it's 8-bit. You can just make it, yeah, any color you want actually. Let's keep it on magenta. Why not? So now to calculate the density of our processes, this is how the binary image looks like. We need to set our measurements from the mean intensity to area fraction. Area fraction is the measure of density. It gives us the percentage of our region of interest that is taken by our object rather than background, so by our processes. To do that, we go to analyze, set measurements, and then instead of mean green value, we choose area fraction. We, again, drag our oval to the region of interest. We then click M, or maybe here, or maybe here, and here. Yeah, so we have all of the regions of our slice probed separately. So you can see that in this particular area, which is in the border between this structure and this brainstem structure, the area fraction that is taken by astrocytic processes is around 25%. Whereas here, these processes are much less dense. So they take about 3%, here about 1%, here about 10%. And again, you just take these results into Excel, unblind them, and then proceed to your statistical analysis. Okay, so I'll quickly come back to the presentation and just to wrap it all up for you. So what we have learned today, we have learned basic things about FIJI. I think the take-home message is the good start is actually to open FIJI and try to do some measurements on your pictures, on the images. I hope know how to draw multiple region of interest and how to measure within our region of interest. We know how to deal with our images if we want to make a composite. So we have all of our colors on top of the others, or we want to actually switch between them. We know how to make a Z-stag or Z project, which gives us one picture that contains all of the information from the whole stack, from the whole depth of our slice. We also, I hopefully touched on the topic of image binarization. And here I'd like to stop for a while and tell you, if you want to do some sophisticated morphology analysis, for example, you want to know how complex your dendritic tree is and use some shoal analysis, that's definitely not enough what we've done today. The adaptive thresholding is going to be your first step, that's for sure, but that's only the first step. And then using FIJI, you can do a lot, but that's probably a topic for the next seminar, because it would take me another 2 or 3 hours to go through all the things you can actually do to make your binary images beautiful and your processes really nicely binarized, and then you don't lose any information of them. And at the end, I've showed you some very simple ways to measure intensity and density of your staining, which I think are simple but really useful when you want to do some quantitative analysis of your slices. And the main conclusion would be that image analysis now is actually essential. Okay, you can present your beautiful picture in your paper and show, okay, this is the antigen, it's present there, but I don't think it's enough anymore. Also, FIJI is your friend. It's a very basic tool if you want it to be basic, but if you want to go fancy, it may become very sophisticated, and I really like this balance. So you can start simple and then you can learn a lot and really dig deep into it. And yeah, of course, it was a very quick seminar. We just touched at the tip of the iceberg when it comes to these topics, but I hope now you've got an idea where to start your learning curve when it comes to image analysis, and now you can actually deep to many more resources that are available in the internet and instruction videos online. Thank you so much for your attention, and I'd be happy to take some questions.
And I'm sure while you've been talking, you haven't noticed, but there have been a lot of questions that have come through in the chat. So I'm just going to choose a few, but I just wanted to say out loud that if you missed any of it, then the recording and the slides will be available to everyone within the next day or two. So you'll be able to watch it again if you want to go through what Lukasz just talked through, if you want to practice yourself side by side with your own images. Sorry, my voice is going. So Juma asked, in your opinion, which option do you choose from for average or max intensity for the Z project?
So yeah, it really depends on your question and your image, but I'd rather use max intensity because when your object is not very dense, so let's say there's just just few processes, and then your slice is very deep, and you've got a lot of background on the other images in your Z stack, then you lose a lot of intensity when you average it. So with analyzing processes, for sure, I would use max intensity. I would most often actually use max intensity for the analysis. However, if you just want to make your picture presentable and really nice, you can pick and choose from these two. And with some colocalization studies, for example, the average intensity may be a better option, but for measuring density and binarization, I would always choose max intensity.
Great, thank you. And then we have another question from Adriana, who said, do you have any recommendations for colocalization with 3 channels of interest, not including DAPI?
I don't know what recommendation are specifically asked for here, but if you just want to do a colocalization study, so we'd like to, for example, count your cells that co-express 3 different things, not including DAPI, then you can probably go with the option make composite and draw some rows, so some ovals around your cells, and then just manually count them. Of course, you can do it with measurements here. You don't have to like do it by hand and manually count them, but you can just draw multiple rows and then make the measurements for all of them.
Thank you. And then another question from Verena is the threshold only applied to all the images that are open at the time?
It's actually applied to only the image that is clicked on, so only to the active window. Even if you have some pictures in a stack or or there are multiple channels at your picture, the thresholding will be done only on the active picture. So, for example, with our staining, we had GFAB in magenta and DAPI in cyan. When I've done my thresholding, it was only applied to the magenta channel, and DAPI was still not binary.
Great. And a couple more questions. So someone has also asked, how do we know what type of thresholding to use? And do you do this before you analyze a Z stack? I think they mean before you create the Z stack.
So first, I always create Z stack. That's how I start. And then I do my thresholding. And again, if your whole image is nicely illuminated, like homogeneously illuminated, you can just do a simple thresholding. And again, check if it looks okay for you, just stick to the simple one. However, if there are some local differences in contrast, I would highly recommend the more sophisticated adaptive thresholding. And also, if you have thin processes, I would also recommend the adaptive thresholding. And I will repeat this again, probably not very popular opinion, not very scientific, but you have to go through a lot of trial and error because it really depends on your image. So try all these algorithms. You can definitely assess by yourself if it's binarized nice or not. If you lose some image, some object, try a different one. If you have too much background, try a different one.
Yeah, and I just want to add to that as we know, biology is incredibly variable. So we've just shown you one very specific example for one type of brain area, but do have a look either online and in the literature to get an idea of what other people in your research area are doing. There are so many really useful like YouTube videos and tutorials and other blogs out there that will help you use FIJI, and they might be from someone, say, in your area that's in your like research area that will help you out. And then I just had one more question, which I don't know if you'll know the answer to, Lukasz, but is there a way to define automated nuclei counting in FIJI? For example, is there a plugin that could be useful?
Definitely. I don't know the plugin at the top of my head, but yeah, definitely. With cell counting, you've got a lot of plugins in FIJI you can choose from. With the binarization, you've got a lot of plugins. Not only these ones that I've shown you, there are plenty. Also, you can record your own macros, so you can, draw multiple region of interest, set your measurements, and then record the macros, and then create somehow your own plugin. So, but I don't know the plugin name from the top of my head, but definitely there are a lot of tools to count your cells.
Yeah, I can follow up with that. You can, if you start to get really confident with FIJI, then you can make your own macro, your own plugin, and save it yourself. And then, for example, you might be able to share it around your collaborators or your lab. I used to do that in the past, and that was really helpful. Again, there are lots of really useful resources online. And yeah, that's great. Also, just want to say,
visit GitHub as well. You can find everything there.
I think that's most of the questions that we'll be able to answer now. A few of the questions are a bit too specific to answer live, but what we'll do is we'll make sure that we answer every question. We'll email everyone that's answered questions because we've got so many. And also, Lukasz, we've got so many lovely comments coming in saying thank you for the really useful presentation. So thank you very much on behalf of everyone.
Thank you so much.
I found that really useful. And I also just wanted to say thank you everyone for listening. Like I said, that we'll have an access to the recording, and also everyone that attends will be able to get a free sample of a protein tech antibody as well. I just have a few other slides that I'll present afterwards, but if there are any questions, please do feel free to email either myself or Lukasz, and we'll be able to answer any of your questions.
We'll be happy to.
Sorry. So I just want to talk a bit about our products that we have available for immunohistochemistry and for immunofluorescence. So first off, we've recently launched our brand new IHC Easy kits. And so these are IHC kits which contain everything that you need for a IHC experiment. And this includes the primary and the secondary antibody, as well as the polymer HRP secondary and DAB staining protocol. And so these are actually the first on the market to contain the primary and the secondary antibody. And so what we've done is we've made sure that the R&D and the manufacturing process means that you can literally just do these and do one run and get excellent images first time. We've got predetermined sample antibody dilutions even, as well as all the antigen retrieval reagents as well. And also, as Lukasz mentioned, he used one of our Correlite antibodies. So that GFAP that you saw was a Correlite 594 conjugated antibody. And so our Correlite antibodies are simply our popular primary antibodies that have been directly conjugated with a synthetic fluorophore. So Correlite is essentially our Elexaflor. If you're not aware, Elexaflor is now off patent. So that means that companies like Protein Tech can actually create their own synthetic fluorophores, which have really similar equivalent brightness and stability like the really popular Elexaflor dyes. And the reason that directly conjugated antibodies are good is because you don't get any off-target binding from your secondary antibody. And so that means that you get much crisper images, so therefore have higher resolution. Would also like to talk to you a little bit about our nanosecondaries. So we sell our nanosecondaries through our brand Chromatech. And so nanosecondaries are essentially nanobodies which have been conjugated direct to a fluorophore. And nanobodies are 10 times smaller than your traditional IgG. They're actually only 15 kilodaltons instead of 150 kilodaltons. And these mean that you get really, really excellent staining because you've got a much smaller linkage displacement. So because the secondary antibody is so much closer to the target of interest than that of the bigger primary and traditional secondary antibody complex, that means that nanosecondaries are excellent for high resolution images as well as super resolution microscopy. As you can see here, this is what I was talking about. You can see this huge complex that you get of like 4 or 5 secondary antibodies binding to your one primary. Whereas when we have our nanosecondary antibodies, even you can see how much closer they are to the target. And these are excellent for super resolution microscopy, such as STORM and STED. Sorry, this is just an example of triple mouse multiplex staining. You can see here in the HeLa cells stained against common cytoskeletal and beta-laminin targets. You can see the really beautiful stainings here. Sorry. Oh. And this is something similar. So this is a nano label. So this is where our nanobody is stained against actin and vimentin. And you can see here in super resolution microscopy, this is a STED image of HeLa cells. You can see how the nano booster, the nano label is applicable to really, really high resolution images simply because of how close your fluorophore is to your target of interest. And so that's it from Protein Tech and from me and Lukasz. Thank you so much for attending. Just want to let you know that everyone will get a link to apply for a free sample today. So you can try one of our Correlite conjugates if you're interested, or you can also try a free sample of our Chromatech nanosecondaries. And I just have one question about Correlite. Is there any improvement on Correlite fluorophores compared to Elexaflor? There isn't much of an improvement. We have done some preliminary experiments at the moment. Our R&D team did, and they've shown that it is a little bit brighter. But again, like I said, they are pretty much the same design, the same formula as Correlites. So as Elexaflors even, sorry. So that's one thing.
The improvement is the price. It's a really nice price.
Yeah, they are cheaper because again, they're off patent, like I said. And Juma just asked, how do you get the free sample? I'll send a link. Everyone will get a link to the free sample at the end. We'll send a follow-up email with a link to this recording, also a PDF of the slides, as well as clear instructions on how to claim your free sample. All right, thank you so much, everyone. I hope you found this really interesting, as I did. And once again, massive thank you to Lukasz again.
Thank you so much.